this symposium. We are very, very excited for, yeah, we see everything. So maybe without further ado, let me quickly introduce you. Okay, so guys, while you're taking back your seats, I will introduce you to Claudia, and your last name I'm gonna pronounce Ctortecka. Is that right?
That is perfect.
Awesome. So Claudia is a postdoc in Steve Carr's lab at the Broad Institute in Cambridge, Massachusetts. For those of you who don't know, the Broad Institute is arguably maybe the institute that produces the largest amount of genomics and proteomics data in the world. Like all of your paper, they have at least 1,000 samples. Claudia actually works there since the beginning of the year, and she works on low input and single cell proteomics. So prior than that, she was a PhD student with Carl Mector, that you will hear from him after her. And in collaboration with the Selenium, she uses this picoliter dispensing robot, the Selenium-1, that she co-developed the proteoCHIP. These are actually milestones in the single-cell proteomic community. So we're all very excited that you managed to connect from California, even though it's very, very early, and we are looking forward to hear your talk.
Thank you, thank you. Thank you for having me. I'm actually very, very excited to present this data to you, some of which is very early under construction, and I'm very much looking forward to a lot of questions and discussion afterwards. And I'm actually very sorry that I cannot be there in person, but we had another single cell meeting here in California. This is also why I'm on the wrong coast, and it's particularly early for me. But so let's get right into it. Today, I will be talking about multiple different aspects of our single-cell proteomics workflow. And as I said, most of them are still very heavily under construction, but we try to find the perfect way by combining multiple different aspects that are right now still also very interesting for a lot of different people and a lot of different approaches. So in the last couple of years, multiple groups have actually started to work on improving sensitivity of current workflows. And they have been finding very, very smart ways how to overcome limitations in sample preparation and also acquisition. And we have therefore currently we do have a lot of different options to choose from, and I will, for obvious reason, today focus a lot on the work that we have been doing together with Selenium, where we have developed what we call the proteoCHIP, and our take on label-free single-cell proteomic sample preparation to most efficiently deliver our specimen of interest into the mass spectrometer, and without losing pretty much all the peptides on the way, which is actually a real issue in single-cell proteomics. So for this, as I said, in very close collaboration with Selenium, we have optimized our label-free sample preparation workflow using their picoliter dispensing platform, the Selenium-1, and the label-free chip that you have already heard a lot about from David. So very briefly, these two come together to most efficiently perform cell isolation, detergent-based cell lysis, protein digestion of our single cells, and ultimately we want to deliver the peptides to the mass spectrometer with as little losses as possible. And we do that by directly interfacing our chips with pretty much any standard autosampler, which also very much overcomes any kind of manual sample handling. So a bit in more detail, we process our single cells in about 40 nanoliters of our master mix, which is comprised of an IMaSS-compatible detergent and our protease of choice. So our workflow actually allows you to combine different proteases or really go for whatever is most suitable for your workflow. And we have automated the entire workflow, basically taking the human out of the equation, which we noticed actually does introduce a lot of variability and errors throughout the processing. And after the previous talks, I don't really have to go into too much detail while we do want to perform label-free analysis, and when we perform label-free analysis, why we want to go for DIA. Obviously, due to obvious reasons, we reduce missing data by overcoming precursor stochasticity, and the increased analysis speed just allows us for much higher throughput compared to DDA analysis. But what I really want to highlight here is that since we aim at regularly isolating and fragmenting all precursors equally across the entire sample cohort, we really needed to widen the isolation windows when performing DIA. And that in turn results in a lot of co-isolation of multiple precursors, making such spectra inherently difficult to identify. But this is exactly where the timsTOF and my favorite, the timsTOF SCP, comes into place. With the added ion mobility dimension, we actually continuously concentrate and pre-separate the ion packages. So when we look here at the extension of DIA to diaPASEF, we actually can see that in the analyzer number 1, while the ion packages are being trapped, we do continuously elute using the analyzer number 2, which you have probably heard already a lot about, and that actually allows us to introduce a third dimension of separation, where we not only separate according to retention time and mass range, but we do actually also have the additional characteristic of ion mobility, where we can see that these 3 precursors on the left-hand side, I think my pointer doesn't really work. Maybe we can do that. Yes. So the 3 precursors that have that would be co-isolated here on the left-hand side in classic DIA are nicely being separated whenever we are performing diaPASEF, and therefore reducing the complexity of the individual spectra, allowing us for a better identification rate. Now, with this in mind, we really wanted to tweak our low input and single cell diaPASEF strategies to be as quick as possible, but also to be as comprehensive as possible. So when we here look at the precursor distribution of a normal triptych sample in M/Z on the x-axis and ion mobility on the y-axis, we can see that the productive area is very distinct, and we developed a dedicated method to cover around 95% of this exact area. And we wanted to keep the cycle time as short as possible to accommodate the short gradients that we're using to improve on throughput whenever we're looking at single cells. So to allow for proper quantification and a minimum of 5 points across the peak, we use 100 milliseconds RAM time, or even lower, with a 35 Thomson isolation window, and that really results for us in the best results. But let's dig deeper into the first question that I usually get from my colleagues, and that is, how will you analyze all of this data, and how will that impact actually my searches and my time on the servers? And our initial option for timsTOF DIA data was Spectronaut. This is very highly used, mainly because also the interface is very practical and the data can be easily explored using their tools. And Spectronaut actually integrates seamlessly their DDA search algorithm Pulsar to generate experimentally derived libraries. And more recently, they have also implemented the library-free search approach, which they call direct DIA. And an alternative would be the famous dia-NN, which is now also integrated in FragPipe to combine MS Fragger for DDA searches and also library generation, and then perform high throughput dia-NN searches. And even though Spectronaut and dia-NN allow for the usage of pretty much any DDA generated experimental library, as I said, as I point out here, the IMaSS Fragger for dia-NN or Pulsar for Spectronaut is the most convenient option right now. And even though FragPipe is actually proposed to run on a desktop computer and TIMS datasets are getting smaller and smaller, we do still encounter a lot of difficulties. And this is especially true for my very sparse single-cell data, which takes ages to run, and we create huge intermediate files. And usually my laptop is not very happy if I have to urgently run something locally. And this is why I was very happy to see that in our lab, Mani and Koi started to implement both dia-NN and FragPipe on Terra. So Terra is a Google Cloud-based platform that is very easily accessible, basically from any browser that you're using. And you don't have to have any local hardware requirements, but you can still massively parallelize all of your analysis. So what I really like about this implementation in Terra is that you can actually share also your data, your pipelines, and also the intermediate files. So whenever you're processing your DIA runs and you really want to use a multitude of different libraries or different kind of parameters, you can reuse those intermediate files, you can share those with your colleagues, and you can save a massive amount of time and also computational resources. And then at the end, you just download whatever you're most interested in and do your post-processing from there. Obviously, this mainly relocates the difficulties, but this is very suitable for impatient people like me that want to basically look at data in real time, which was implemented by Broker in their Pacer box. So their Pacer box started out with allowing you to analyze DDA data on the fly. And that already improved my experiences in extensive method optimization. But most recently, they have extended their platform to library-based DI searches on the fly. So for this, one can use integrated their integrated ProLucid DDA search and upload pretty much any spectral library. So while acquiring your DI files, all are individually searched and then can retrospectively be integrated to match between the sample sets, which brings them up to speed to any of the other two options that I was introducing to you just now. And the Pacer runs, as I said, on the Pacer box, which doesn't occupy any of your computational resources. And it only requires the addition of a computer that is basically placed right next to the timsTOF. And what I nicely also learned last week can actually be checked from your phone, which is not particularly necessary, but it's great for people like me who actually manage to have all of their instruments usually fail 3 seconds after you're leaving the room. Anyway, let's look into the data. So I have, for this very detailed example, I have created a library from single-shot DDA injections for label-free single cells, which are prepared exactly like the dia-PASEF runs that we're now looking at. So the DDA runs were searched with ProLucid, and I used the exact same library for all softwares to enable a back-to-back comparison. We additionally also created libraries with Fragger and Pulsar, and we also searched them, and all of them really showed the same trends, but due to time constraints, I'm not going to go into too much detail. When we now combine our dia-PASEF method, our label-free proteoCHIP sample preparation, and acquiring the data on a timsTOF SCP, we do reproducibly identify around 1,000 proteins per single cell injection when we use the single shot DDA spectral library with the newest version of Spectronaut and dia-NN. What I also included here in light green is an earlier version of dia-NN, which is 1.7, and that is actually the basis for TIMS dia-NN. So we can see that Spectronaut and the newest version of dia-NN 1.8 perform perform very similar. And the earlier version of DIA-NN 1.7, which is, as I said, the basis for timsTOF DIA-NN, also do deliver very similar results. But important to note here is that I'm actually looking at the first pass results, if anyone is familiar with DIA-NN. So this, you have multiple steps of analysis, and I want to point out that it is really important to have a back-to-back comparison. Now, when we look at the missing data across runs, which I find for these very large cohorts extremely important to really look at data quality and also comprehensiveness across runs. Where I also want to make clear that if anyone has seen my previous presentation of this data, I am here showing the exact same data, the exact same analysis, and pretty much also the same conclusions, just displayed in a very different way. So we can see on the x-axis here the number of analytical runs, and on the y-axis the percentage of missing peptides across DIA-NN in green, Spectronaut in red, and timsTOF DIA-NN in blue. And what we can appreciate is that most peptides are actually not found in all of the analytical runs, but overall they're just missing in a couple of those. And all of the missing data is pretty much is equally distributed across all three softwares. However, what we can see from this drop here at the end of the curve, that both DIA-NN and timsTOF DIA-NN outperform Spectronaut in precursor recovery, where we can also see that timsTOF DIA-NN actually performs slightly better even than DIA-NN, where they integrated also the collisional cross-section prediction as improvement of recovery. So this dip in the curve actually reflects around 3% of all precursors that are better recovered using timsTOF DIA-NN, which represents about 200 peptides, which is not a lot, but still, if you go for these very sparse data sets, that can also be very, very important to recover those concrete profiles. Now, we wanted to have a look at how accurate these identifications are and how well they actually compare in quantification. And when we look here at these different DIA-NN on the x-axis and Spectronaut on the y-axis, where we have here also Spectronaut versus timsTOF DIA-NN, and then the final comparison of timsTOF DIA-NN and DIA-NN. We can see that actually the quantification is very comparable at using all of the three softwares. However, what we also see is that the trend of less comparable and less correlative is towards the lower abundance, which is not very surprising to us, but still a very valuable information when we're trying to validate and evaluate our data quality, and also which software is most suitable for our data analysis and whatever we want to really achieve with these data sets. So how do we actually, after all, check for false identifications? And for this, based on a very great discussion that we had after my last presentation at ASBMB, we generated not only experimental derived libraries, but also we wanted to look into predicted libraries to really do estimate the FDR, the actual FDR of these search algorithms. And we tried to trick basically the software in making mistakes. And to look at these different approaches with different library sizes, we generated the standard approach, how it is actually recommended to use with the standard human FASTA file. So that would be the first option, where the software actually generates the decoys based on their pre-filtering and scoring parameters. On top of that, we created a library including 50% of razor peptides prior to decoy generation. This basically inflates the library size, but only leaves us with around 1/3 of spectra in the library, which are theoretically matching to whatever we're analyzing. And lastly, we combined a fast dia file with a decoy organism that, in theory, should result in very similar results as our decoys that we have generated computationally, but we can really look at how many of those identifications that we find actually do not overlap with the human sequences and are supposed to not be identified. So let's compare. We here have only used Spectronaut and DIA-NN and did not include timsTOF because it's currently still under heavy construction to use these very large libraries and also the predicted libraries. And I hope that very soon I will be able to include timsTOF into this plot as well. So just to make sort of as a sanity check, we searched only our non-human FASTA file, and we did not find any identifications when using DIA-NN, and only a couple of peptides when using Spectronaut. So that was already great. However, when we then looked into the different softwares. So first of all, we have the normal setup where we use our human FASTA file and the prediction. We see that the identifications from Spectronaut are very similar to our experimentally derived library. And with DIA-NN, you actually do see an increase of 20% of identification when using the predicted library. And this is even though we actually search against a extremely large library. And by extremely large, I mean an 8-fold increase in size. And still, these two softwares can handle these extremely large data sets very well. And now when we inflate the library size twofold by actually performing these decoy searches, we do lose identifications of 7% in Spectronaut and 8% in DIA-NN. But this is very comparable to also the combination of the human and non-human library. However, when we then look at the actual FDR that we really wanted to evaluate, we were actually surprised to see that Spectronaut results in around 0.1% false peptides, whereas DIA-NN actually does identify over 3% of actual false peptides. We currently do not know, so we do not have a concrete answer why here Spectronaut is actually yielding such great actual false discovery rate over DIA-NN. As I said, this is very heavily under construction, and we try to represent both algorithms as best as we know. If you have any questions about parameters that we were using or any kind of detailed questions, I'm happy to share all of our parameters that we're using, and you're happy to contact me afterwards if you have detailed questions about this. But what we were also interested in is actually the overlap of these two search algorithms. Now, when we look at the strip peptide sequences, which basically represent peptides without any modifications, I have been showing at my last presentation of this data set that the current version of DIA-NN and the current version of timsTOF actually results in 45% of overlap when looking at peptide sequences across these very sparse data sets. And funnily enough, also when we use predicted libraries here in this case, comparing Spectronaut and DIA-NN, we again see a 45% overlap of peptides that are identified. This is not something that everyone would like to see, but this is the current state of analysis. And after the attendance of the last conference last week, we have seen actually that a lot of people see very similar results from their single cell and very sparse low input data. And we're still in the process of evaluating what gives rise to this reduced overlap. And we started by doing a comparison of actually dynamic range. And what you can see here is the intensity of peptides that are identified and quantified using Spectronaut and DIA-NN. And what we see was actually reassuring and really nice to see that both of them do span 5 orders of magnitude. And also the overlap actually spans 5 orders of magnitude. So it is not particularly a matter of abundance or, for example, Q-value annotation or the number of matched fragments in these profiles. But if you have any suggestions on what those reduced overlap could arise from, I will be very happy to look into that and hopefully include that into my next presentation. But all of this sort of leaves us, I don't know if it's confident or inconfident that recent developments in library-free searching actually allows us to compensate for the lack of a deeply fractionated library. But we do hope that these library prediction actually allows us to better represent biological diversity. As we have just heard, the definition of a library and is highly important to really represent whatever you are looking at. So you really want to recover as best as possible the diversity that is represented in your sample. And we currently think that this is possible in the best way using these predicted libraries. And this also reduces actually the time that you need for library generation, and therefore also your final throughput. And that will bring me to my last very short topic for today, is how we not only want to push the boundaries and in throughput when performing dia-PASEF analysis, but we actually do also want to focus on chromatographic throughput. And for this, we have once again worked very closely together with Selenium on a dedicated proteoCHIP design that can be interfaced with the famous AVASEP that currently allows us to operate on an 80 samples per day method in their nanoflow mode, the Whisper. And I'm very happy to share with you some very initial results using this AVASEP proteoCHIP and our optimized dia-PASEF strategy that allows us actually to increase our recovery of peptides around 50% by directly connecting our sample preparation with the AVATapes without any kind of manual sample transfer. And I'm sure that Carl will elaborate in a minute how important overcoming of sample transfer is. And so we do here identify around 1,000 protein still using the 40 samples per day method. And very, very preliminary data. We have started to subsidize acquisition time with dia-PASEF and the newest generation of Aurora columns, which are only 5 centimeters long and allow us to process, as I said, 80 samples per day. And we're very happy to see that we are actually covering the exact dynamic range of samples when we use our timsTOF SCP. We're still very much working on together with AVASEP and INoptics to optimize our separation to really recover similar identification numbers as we do see from our 40 samples per day method. And I hope that very soon I will be able to complement that with more updates. And concluding, I hope that I was able to show you today that there's actually a multitude of different approaches out there. And obviously, I have only touched upon our proteoCHIP-based workflows, even though these are, there are a lot of alternatives out there, and all of them have their own very interesting advantages. However, our dedicated label-free chip right now allows us to process very sensitively large cohorts with good, acceptable data completeness using DIA on the timsTOF SCP. And I would like to also point out that the chromatographic throughput, especially with the direct connection of our chips with pretty much any standard auto sampler, is massively improving peptide recovery. And lastly, that also the cloud-based implementation of DIA-NN and IMaSS, as well as the real-time PASEF analysis, makes our lives a lot easier, but I think there is a lot of high demand out there in terms of data analysis and data validation, and I'm sure that there will be a lot of great tools out there in the near future. And I hope that I could give you a brief overview of what we're doing, and as a very short outlook, I'm very happy to share with you that, and also to see that people are actually interested in that, as other people are interested in that as well, is that we're extending our workflows now to spatial resolution, and most importantly, with the long-standing expertise of the Broad Institute in regard to PTM work. And before I jump to my most important slide for today, I still want to add that, in my opinion, there is no perfect workflow, but rather the need for a multitude of different approaches tailored to the individual project and biological question. So I think we all need to work together and really push the field forward. And with that, I'm at my acknowledgements, where I would like to thank, first and foremost, obviously, Stephen Namrata for giving me a new home at the Broad, and work together with such amazing people, all shown here at our lab trip to our very fancy steakhouse in Boston. And I would also specifically want to highlight Mani and Koi, who spend an awful lot of time fixing everything that I crash on Terra. Definitely also Carl for introducing me to the single cell field and starting these terrific collaborations with Selenium, where I want to highlight David, Angeli, and Guillaume, and of course Brooker for their continuous support, specifically John from the PASEF team, and Seb, who started out with me at the Broad and taught me pretty much everything I know for the timsTOFs. Lastly, Dorte and Nikolai from AVOSEP, and Jared from Ion Optics for getting me started with their very high throughput. And with this, I'm very happy to take any questions. And I hope that I will actually be able to hear you.
Oh, can you hear me?
Yes, I'm just cranking up my volume.
Thank you very much, Claudia, for this great presentation and for finding time this early to connect with us. Questions from the audience? Oh, I think your ex-boss has a question.
Good morning, Claudia.
Good morning, Todd.
Yeah. Really great presentation, like it. The only question what I have, at the end, you optimized a lot of the methods with the nanoelute, and then later you switched to a type of other HPLC with a method it's called 40 or 80. How can you transfer the accuracy and the precision of the data with the 40 or with the 80 method? And I was not sure how many proteins you got with the 80 method, end of the day. I think you did not really show it up, or I did not see it.
Yeah, so that is an excellent point. What we can achieve with the nanolute is very comparable to what we see on the 40 samples per day method. We really wanted to push the boundaries there, where we really want to push on throughput to move towards the 80 samples per day method. And as you usually do before going to a conference, you start to acquire data last second. And I did that prior to leaving for California. And we have, as I was showing, we can recover a very similar dynamic range using the 80 samples per day method, but our identifications drop by about 40%. We're still actively working together with Ion Optics and AVOSEP to really optimize this method, because the 80 samples per day method is not yet officially released. And we do know that our chromatographic separation is not perfect. So we did tweak a bit on the acquisition speed, and I hope that we will push identification numbers as well to be in the range of around 1,000 to even higher identifications when using the 80 samples per day method.
Thank you.
More questions? Can I ask a quick question? So Claudia, actually, you blew me away when you showed we have 5 orders of magnitude in protein quantification, which is honestly very, very similar to the dynamic range that we get when we use like infinite amount of material.
Yeah.
So usually when we inject a lot of material, I explain it to myself that the very intense ions, they steal ionization from the low intense, they basically steal capacity of the instrument detector. You don't have this problem clearly when you do single cells, but still you get the same dynamic range. How do you explain that? I mean, I don't know if there is an easy explanation.
Yeah, so we think that actually the limitation of this dynamic range is very similar to when you're dealing with high input material. So when you're dealing with high input, you're basically saturating and you're competing for analysis time with the very high abundant ones. But when we do the low input injections, we do not, so we think we do not have sufficient ions, and we have to spend an awful lot more time on equivalent dynamic range to represent this dynamic range. So you're not overloading, but you're underloading, which means you're cutting off at a different area. So I think to really extend this dynamic range further, there's a lot that need to be done, but I think we're moving into a good direction where this actually will be possible, that we can concentrate the ion packages even further and do extend the dynamic range lower.
That's great, thank you. Any last question until we move to our last speaker? Okay, we'll move to our last speaker. Thank you so much, Claudia. We'll let you go.